It's all right. So, once again, welcome all of us to this amazing webinar series. This is the African Science Network, an international, globally recognized, impactful organization striving towards uh, the propelling of innovations, science, networking, collaborations all across African researchers. And the African Science Network has currently over 7,000 membership in various platforms, especially particularly the WhatsApp group. And that platform has the composition of various distinguished personalities, vice chancellors, directors from all across Africa. And one of the main impactful session of the African Science Network is organizing the monthly webinar series. And that's the reason we are all here today. So, on behalf of the entire admins, the administrators of the African Science Network, I welcome us all today. I'm yours sincerely, Watzani Dauda. And so before we have our amazing guest.
Hello.
Yes, sir.
Yeah, hello, can hear you. It looks like
Hello.
We can hear you, sir.
Oh, sorry. I have something. Okay. Thank you very much, Dr. Wadzani Dauda, for a nice introduction of the AfriScience Network. My name is Aminu Shitu from the Faculty of Veterinary Medicine here at the Usman Danfodio University, Sokoto. Today, we welcome our speaker for the 20th webinar. I call it a special webinar. It is the 20th webinar and on the 20th of the month. So I'm going to go ahead and read the citation of the speaker without wasting much time. Dr. ThankGod Echezona Ebenezer was a bioinformatician at qbio.ai and the University of Cambridge School of Clinical Medicine, where his role focused on addressing drug discovery challenges using AI and developing pipelines for early cancer detection using circulating tumor DNA methylation markers and machine learning techniques, respectively. Prior to joining Cambridge, Dr. Ebenezer was previously a bioinformatician at the European Bioinformatics Institute, EMBL-EBI, within the UniProt team, where he developed computational pipelines for automatic functional annotation of proteins using evolutionary models and software engineering best practices. He undertook his postdoctoral research training at the Ilham Institute, EI, a UK national facility for genomics, where he worked on wheat transcriptomics and epigenomics. Dr. Ebenezer studied for his PhD degree in biochemistry at the University of Cambridge, UK, where he worked on the genome of Fugulina gracilis. He has a MSc in environmental toxicology and pollution management from the University of Lagos in Nigeria, and a BSc in parasitology and entomology from Nnamdi Azikiwe University, Awka, in Nigeria. Dr. Ebenezer applies software engineering best practices in pipeline development for big data in biology, such as genomes, transcriptomes, epigenomes, and proteins. Dr. Ebenezer is involved in fostering dynamic international and regional collaborations in science. In 2020, he founded the Euglena International Network, EIN, and the African BioGenome Project, AfricaBP. Dr. Ebenezer led the publication of the AfricaBP vision in two science journals, Nature and Nature Biotechnology, respectively. In 2023, AfricaBP trained over 400 African scientists in hands-on genomics and bioinformatics practicals using local African resources, and brought together nearly 4,000 African scientists across 48 African countries through a series of Africa-based workshops involving 24 African organizations. So today, Dr. Ebenezer will share his insights on the African BioGenome Project, AfricaBP, progress and perspective. We are privileged to have him here to enlighten us on the advances and aspirations of these groundbreaking initiatives. Before I hand over the floor to Dr. Ebenezer, I would like to inform you how the meeting is going to be. Dr. Ebenezer is going to speak for 20 to 25 minutes, and then the rest is going to be interactive session. So we will be discussing, exchanging ideas and thoughts, and asking questions up to the end of the webinar. So, ladies and gentlemen, please join us in warmly welcoming Dr. ThankGod Echezona Ebenezer. You're highly welcome, sir.
Thank you, Dr. Aminu Shitu. So it's great to be here, and also thank you for inviting me. I think we've been talking for a while now. Also, Dr. Wazeni, and for anchoring this, and to all the distinguished scientists and researchers on this platform. I'm really pleased to be here, and today I will talk to us about the African BioGenome Project. So I'm going to share my screen. I understand I would be able to talk for an hour, but I normally prefer talking for 20 minutes and then interactively, diving deep into other things without presentation and having like an interactive session. So let me share my screen. I hope I'll be able to share my screen. So can you see my screen now, please?
Not yet. Okay, it is coming up.
Okay.
Yeah, we can see it now.
You can see it now. Okay, I'm going to put it in presentation mode.
Okay.
So let me see. Just let me know, because sometimes this doesn't work all the time. Can you see it?
Yeah, it is working now.
Okay, all right. Okay, thank you very much, distinguished scientists and researchers. My name is ThankGod Ebenezer, and I am going to talk to us about the African BioGenome Project, which focuses on the future of biological diversity across Africa. I'm also speaking on behalf of my colleagues at the African BioGenome Project. So just a bit of a content, I am going to talk to us about the background of the AfricaBP, the strategic goals, the key highlights, the emerging opportunities, the sequencing efforts so far, and also what the emerging regulatory hurdles in genomics would be in biodiversity, or are in biodiversity. So a bit of a background and goals. We all know that the global biodiversity is happening at an unprecedented scale, and we need a huge urgency to be able to reverse this trend. And we must be able to protect Africa's biodiversity. And this diagram shows the various, the parts in red shows where the biodiversity is currently being depleted, and Africa is not left behind. The part in blue shows where, there isn't much depletion yet. And one of the things AfricaBP wants to do is to be able to conserve and protect Africa's biodiversity through genomics, and ultimately to be able to ensure equitable and sustainable use of Africa biodiversity through genomics. So how do we want to do that? We want to ensure that by being able to achieve the global biodiversity framework, for instance, and being able to maintain the genetic diversity, we'll be able to ultimately achieve this goal, and also help Africa to be able to actualize a third goal in the Komi-Montreal Global Biodiversity Framework. So I'm going to talk briefly about that. And that third goal is the issues around access and benefit sharing, and how do we, through scientific exchange, be able to achieve some of these things. So AfricaBP aims to cover all eukaryotic lineages. Eukaryotic here meaning anything that is not human. So we are focusing on sequencing plants and animal species, and also eukaryotic microbes, as you can see here, plants, insects, algae, and other animals, protozoa, fungi. We don't sequence human, partly because we want to focus our attention on Africa's biodiversity, and there are so many human activities or sequencing going on the continent. But where the gap is in Africa is in our biodiversity. We don't know much about them. There are so many plants and species in Africa that are yet to be discovered, that are yet to be sequenced. Most of them serve as food for us. Most of them serve as cultural, they place cultural significance. Most of them place scientific significance and also conservation significance as well. So here also you can see the vertebrate species, the invertebrate species that we're also looking at in general. So what are the goals of the African BioGenome Project? So the first goal is to coordinate all non-human genome sequencing initiatives. So what we do is that we work with several institutions and scientists in Africa, help them to initiate the genome projects and get it onboarded in their institution, and then they work with us to be able to deliver it. And that could be new project or even existing one, because one of the problems Africa faces is access to the right skills, access to the right people, access to the right equipment, access to the right network. So we believe that by doing this, that we can be able to ultimately solve so many problems on the continent. And we also act as a forum for the non-human space. So that means anything not human, mainly plant, animals, eukaryotic microbe, we kind of act as a forum for that. Then we initiate, develop, and support new efforts, basically new non-human genome sequencing efforts. And we aim that by doing so, we can close the capacity gap across Africa. So thirdly, we aim to deploy this activity, goal 1 and 2, to be able to contribute to regional activities, be it, for instance, the African Union regional agencies or any plant or national institutions. For instance, in South Africa, we have scientists now through AfricaBP that are sequencing genomes at their national agency, and then working with AfricaBP to be able to understand what are the pipelines to use for the sequencing, what is the best practice, and we also provide support for them, and then that contributes to the national science agenda. So AfricaBP started, actually I wrote the proposal in 2019, and then we started discussion in 2020, but in 2021 it then started properly. We have 5 grand challenges. The first one is genomics and bioinformatics technologies for the agri-environment. This basically focuses on anything technology, be it genome sequencing technology, bioinformatics tools and processes. Then the second one focuses on crops and livestock improvement and health. This is more around the, things like the genetics of the crops or livestock and how that can be used to improve it. So it's more around translational aspects. Then there's also the genomics for conservation of endangered and endemic species. Then the fourth one is because biodiversity in Africa and anywhere around the world is not owned by one single person. Most of the times it's caused across communities, because even though it's found in your garden or in your backyard, it does not often belong to you. Sometimes it belongs to the state government, to the national government, and things like that. So we also understand the socioeconomics and the ethics and policy issues that surround that. So we have a grand challenge that deals with that. Then the fifth one is technology development, knowledge exchange, and industry. So one of the things we aim to do with AfricaBP is to ensure that there's cross-pollination of experiences, of ideas, and of just about anything, that we don't have to do things on our own. We bring everyone together. So I'm going to show us in the next few slides how we've done it. And why there's that cross-pollination of ideas and experiences, one other things we aim to do is that we also provide a platform for this to provide, for instance, technology development opportunities. So how is the project currently being funded? So we are working on potentially basically 3 funding mechanisms. So we leverage on global opportunities, carrying out linkages between global network and also African institutions. For instance, the vertebrate genome, we're able to link up the vertebrate genome project with the University of South Africa to fund several species, and then that then provoke additional support and investment by the university into their own institutional science agenda as well. So we're also leveraging on technology companies. For instance, I'm going to show in the next few slides in our publications, where we also receive support in order to sequence active species and also to conduct, for instance, trainings. So what have the project achieved since 2021? I must mention here that the project is mainly led by young African scientists and also established African scientists. And everyone is, putting in their effort and then driving things at the national level, at the regional level, as well as the continental level. So because we believe on the idea that if we can get things sorted at the local level, then that could potentially reflect at the regional level. So we launched in 2021 and published a position paper in Nature in March 2022. So this is the position paper which I led, and then that also was featured in the Science Magazine journal, as well as looking at what this could be. And the position paper was featured in more than 100 news or media outlets across the world. Then, as part of the profile of AfricaBP, we were invited to speak at the UN General Assembly, the science session in 2022. I was also invited to speak at the Food and Agricultural Organization about the project and its progress. We're invited to speak at the African Union, and also I spoke about it at UNESCO meeting, and of course at the US Africa Leaders Summit in 2022, and I also spoke about it at the World Home Trust workshop a few months ago. So we have 4 programs in AfricaBP to address the current challenges. The first program focuses on animal species. As I mentioned previously, we only focus on plant and animal species because these are where the gaps are, and also eukaryotic microbes. Then we have a second program on plants. Then we have a third program that is more around ethics and social economics and policy issue, which we call Program 3. We call it Community of Parties on Biodiversity Framework. I'm going to talk briefly about the framework. Then we have a fourth program, which is the Open Institute for Genomics and Bioinformatics. So this is our knowledge exchange program, and this is where we do our regional workshops and kind of also ensure that we create the next generation of leaders across the continent of Africa. The program 1 and 2 are more technical, so this is where the actual genome sequencing effort happens, and several PIs are working on this. Then program 4 is around knowledge exchange, regional workshops, trainings, and program 3 is around ethical issues. So we also have provisions for other opportunities that might come in. So across this, we have things around coordination, fundraising, partnerships, and policy engagement. The project started quite in a reverse order. In most projects, you have the institution, and then you kind of have the project. But in this case, it started like a grassroots movement. And we then in 2022 we institutionalized the project as partnered with the Science for Africa Foundation, and then this was the meeting that was held to be able to institutionalize the project. Then I'm going to talk mainly about the Open Institute and also about projects program 1 and 2. So the Open Institute, as I mentioned, is a knowledge exchange platform, and we organize workshops, and that also include awareness building workshops, providing genomics access to African scientists. We don't have, for this kind of workshop you have to apply, and then during the regional workshops, and then you can be selected. We published the proceedings of this in 2 papers, Nature Biotechnology in 2023 and Nature Genetics. And all of these papers have over 95-97% African scientists. So especially with this, the Nature Genetics one, there are 110 authors, and I think 103 or 4 are Africa-based scientists. Then this is also the regional workshops we held last year. So as you can see, we trained over 400 African scientists. These are scientists with Africa affiliations. And then nearly all the countries in Africa were represented in that workshop, around 48 countries. And we also had people from other countries that attended the workshop. For instance, you can see from China, from Europe, from Latin America, South America, and the rest. And this is the paper of the previous one. So this is in addition to that, the Open Institute also has like fellowships. So we work with African institutions and international partners to grant fellowships. So early last year or so, last year, we launched the Africa Genome Center AfricaBP fellowships. And then what happened in this fellowship is that we had like 10, we were able to select 10 African scientists. So I think in total there were like 600 applications. We were able to select 10 African scientists, and then they spent 2 weeks at the African Genome Center in Morocco sequencing an African species. Then for the fellowship, the leadership fellowship, this one, we had around 200 or 300 applications or so. And then we will select 4 African-based scientists. They are currently in the US now, spending time learning about team science, learning about being a leader in genomics and bioinformatics. And of course, this is done in partnership with the Carl Wells Institute in Illinois, and also with the Mohammed VI Polytechnic University in Morocco. So what have we done so far in terms of sequencing? So as I mentioned, we work with African institutions and collaborate with them and help provoke action at the institutional level. And by doing so, they can be able to sequence their genomes and have ownership for those genomes. So this is an example of some of the species that have been sequenced locally by African scientists and African institutions. And I'm also pleased to see that this is driving change. On the continent, for instance, several of these institutions have now started to create their own genomics program just by partnering with AfricaBP and being able to see the benefit and the significance of sequencing your own genome. But you can't do that if you don't have the right network, if you don't have the right people, if you are not connected, or if you don't have someone that can connect you to the right pipeline and kind of point you to the right direction. So that is one of the big things that we provide, and that is one of the things that one of the gaps that exist across the continent. So we sequenced in total over 20 species locally. We don't sequence African genomes outside Africa because we believe that by sequencing the genomes on the ground or locally, we will maximize the benefits that comes with it. And for instance, your institution and your vice chancellor will know about it, and it will drive change locally. Your national government will know about it. It will also drive change nationally. And then it will build the right capacity. It will also ensure that the students are able to know the process, because sometimes with genome sequencing, people will say, oh, I want to send it outside, you sequence it for me. But I sequence it for you, but you don't know the whole pipeline that goes into it. And then that knowledge is lost. And then not just the knowledge about the capacity, the knowledge about the species is lost. The significance and the translational potential about the species locally in your region is also lost. So that is why we focus all of this are sequenced on the ground in Africa. So emerging regulatory hurdles. So as I mentioned, unlike most taxa, biodiversity is not, you can't say you own this biodiversity, but it's a sovereign national thing, in terms of, it has national ownership. So, and there are, even though you also have like local people at the national level, there's like local people that own it. Most of the time you have, it depends on the regulations that exist, but it's not, you can't say, oh, this is my plant and things like that, because it's shared across border. In fact, even sometimes in Africa, you have species that move from Africa to Europe, let's say North Africa and back to Africa. So at that point, you can't even establish it. So there are several protocols that does that. So we also have, for instance, the Convention on Biological Diversity, the Global Environment Biodiversity Framework, and this is us here talking about this in the paper. We published a recent preprint now that talks about the era of this genomics, biodiversity genomics and bioinformatics in the era of the Common-Monetary Global Biodiversity Framework. What are the ethical, legal, and social perspectives? So because what that happens is that we talk about, sampling species, what we need to do with community, we work with community, look at what are required, the paperwork that are required, and then ensure that the paperwork are gotten, the permits, everything. You cannot just take sample and then go and sequence it. No, and sequence it and then publish it. In this paper, we also mentioned that we have the ethics committee, and whereby for each of the publication, they go through it and then ensure that all the paperwork are fine, and then you cannot just publish it and then the people that own the data or that own the genomes or the samples are not even authors, and so many of the ethical things. So I would also encourage us to read this paper if you have time. So I think with this, I would like to thank us for listeners. As I mentioned, my presentation is short. I usually make it very short and then have more time for presentations. So there are several people on the project, over 100 Africa-based scientists. And these are the names. And also there are also several partners on the project. If you go on our website, you will see so many of those partners. So with this, I would like to thank us for listening. And I'll be happy to answer any question you have.
Thank you very much, Dr. ThankGod Ebenezer, for a very brief but well-encompassing presentation. Thank you so much. And I think I will open the floor for question and discussions. And I have Benjamin here raising his hand. Will you go on, please? You should unmute yourself, please, before you speak. Yeah. Hello, Dr. Benjamin.
Okay. Thank you, Dr. Amin.
Okay. Yeah.
Dr. ThankGod, good afternoon.
Good afternoon, Ben.
Well done. Good presentation. As always, very specific, straightforward, and short. My question is, I'm interested in some of the pipelines you develop in the sequencing. Do you have like a repository where you deposit them for some sort of replication purposes? Like if I want to model a particular project and I intend to use some of the pipelines you developed as a guideline, do you have like a repository? Then secondly, do you have a mechanism on ground for sample collection across Africa? Like some of these species that has been collated and sequenced already, what mechanism do you have on ground that covers the area of sample collection? I don't know if the question is.
Yeah, it's clear. Thank you, Benjamin. So in terms of the pipelines, we have a Git repo, though it's not public yet because we are working on our own internal pipelines. But there are also other external pipelines we use. For instance, with, let's say, already established pipeline like the VGP pipelines that are open source. We also use that. But for our own pipeline, it's the internal and the teams are currently working on it. And there's a Git repo. And what we aim to do is to make it open. Because at the moment, what we do is that we work with institutions and provide those support and the pipelines to them. And once it's all tested and works very well, we'll now make the Git repo open. In regards to your second question, we work with institutions, as I mentioned, that our aim, one of our aim is ultimately to have several like sample ranges potentially in the future. But we don't really think Africa needs that yet. We work with institutions. So let's assume I just mentioned University of South Africa, other universities, Biometrics University. We work with them to get their sample. So they are the ones that do the sample ranging because they are closest to those locations. It is their country, it is their nation. What we do is to provide the right support, the right, you can call it mentoring in quotes, and the right networking and the right, what are the best practices and what are the best guidelines to follow. And then the institutions then arrange their own sampling activities and then get the samples and then work with AfricaBP to sequence and analyze it. And we also ensure that the sequencing happens in the country where the sample come from, because we discourage sending the sample outside. First of all, sending the sample outside Africa, and the only time we send the sample from one African country to the other is when, for instance, there is no sequencing facility in that country. But so far, I think it's only maybe one country like that, because we normally use like a PacBio sequencing platform. It's called the PacBio Review. It used to be called the PacBio Hi-Fi. And if it's not available in your country, then we need to look for the next country closest to you in Africa that is available. So to answer your question, that is an activity that the partner on the ground locally takes care of, and most of them have sequencing rangers. So I don't know if you saw our newsletter recently. In the newsletter, we talked about several institutions that also go out to do their own sequencing, their own sampling, rather.
Okay, thank you very much. I have Dr. Habiba Atta. from ABU, a microbiologist. Dr. Habiba, could you unmute
Okay.
yourself? And yeah,
Hello,
thank you.
everyone.
Yeah, hi. Yeah, welcome.
Thank you. Thank you so much for the presentation, Ebenezer. I'm
Thank you.
actually following your work on social media, and I'm also part of your mailing list. So great work you're doing. Thank you so much.
Thank you, Dr. Abiba.
Yes. So my question is, I follow the work of IPBES and CBD and Comi Montreal Global Biodiversity Framework. And I noticed that sometimes there tends to be like a gap in data from Africa, particularly West Africa. So particularly when it has to do with invasive alien species. So I'm just wondering, in the past few years, I've done some work on plant species and what have you. Has there been any breakthrough regarding the kind of sequences you've gotten that could maybe lead us to think that we have some invasive alien species that could affect biodiversity? And if you found anyone like that, has it been captured as part of the data that is being used by any of the biodiversity frameworks? Thank you.
Yeah, thank you, Dr. Abiba. So we haven't sequenced any potentially or classified invasive alien species, but this is likely to come mainstream as the sequencing goes. You know, as I mentioned, the biodiversity in Africa is huge, with going gradually, and then institutions will sequence and will get it published, and it's going to be an iterative process. But ultimately, we aim that the data we get through AfricaBP would influence policy. So I shared in my presentation one of our publications that focuses on the CBD framework, and again that is also a policy paper in that direction. So as we get more information and get all of those things, our aim is to share it such that that can also support and help the African continent.
Okay, thank you so
Thank
much.
you very much. Okay. I have a question, Dr. Ebenezer. Personal, yeah. I'm interested in cow welfare and udder health. That is the health of mammary gland of cow. There are a lot of things going on there. So for a particular breed of cow, I'm interested in looking at the genetics of that cow. But whatever I'm doing, I don't like just doing things for the sake of publishing.
Yeah.
But I'm interested in application now. So how can I collaborate with the AfricaBP in working on this species, and especially focusing on the aspect of udder health, and also in terms of policy recommendation, as I mentioned, how can I improve this breed? Health-wise.
Yeah.
I'm a vet, so I have to be biased on the health of this breed of animal.
Yeah, thank you, Dr. Aminu Shitty. So
thank you.
We deal with animal stock quite a lot. Actually, one of the partners have a paper on that is potentially still being developed. Actually, several, not just one paper, several as part of the genome sequence for one of the breeds of cow in their region. So we have one of our grand challenge focus on technology development, and that recognizes translational work. Just as you mentioned, how do we go from the basic work and then applying it and solving real problems? So, and to help with that, one way you can collaborate with us is by sending me an email, and then we can get you we can get all the paperwork required for such a partnership with your institution established. Then what will usually happen is that we would be able to link you up with other institutions in Africa or outside Africa that could potentially work with you to deliver that. But before we can get to that extent, we need to see the proposal you have in mind. We need to see the feasibility, and then see that it's actually something that is doable, for instance, and also potentially see where we can support you or work with you to get the resources to be able to deliver that. Did that answer your question, Dr. Aminu?
Yeah, that is very interesting. Thank you very much.
You're welcome.
Yeah. Next person on the line is Dr. Odunayo Aziz. Please kindly unmute yourself and speak. Thank you.
Okay, thank you. Can you hear me now?
Yeah,
Yes.
I can hear you, please.
Clear.
Okay, yeah, thank you for that very fantastic presentation, and I really appreciate you for the work being done. Just like some mentioned earlier, there seems to be a gap in the number of species of animals or any study in some part of Africa, like West Africa and Northern Africa. But I know that we have several individuals that are doing in the personal work and on characterization of local indigenous organisms or animals. Is it possible to curate data from individuals, maybe who have peradventure done something in that area, and assist in bringing some of those things to light? Although it was probably done personally, I mean, by those by individual authors on local, maybe local characterization, local animal species and things like that. Because I think if we can widen that, it will help to bring in so many other species that you have not actually worked on, or that others are yet to work on. So that's my question.
Yeah, thank you, Odunayo. So we recognize that Africa has a huge gap. So I just posted our Nature position paper, which I published. And in that position paper, you can see there's, if you go all the way down, you can see the figure 1, as at that time, shows you the number of genomes in Africa versus number that has been sequenced and number that has been sequenced in Africa, for instance. So we recognize that gap exists, which is why AfricaBP was founded, and to potentially be able to close that gap. And given how large Africa biodiversity is, no single person can sequence it alone. No single institution can sequence it alone. No single country can sequence it alone. So it requires multi-stakeholder, multi-partner, multi-institution approach. So which is what we are deploying. So in respect to what you mentioned, we do know that there are institutions that have maybe local things or one form of activity ongoing. But one of the things also we can't know unless we can have like two things. First is unless it's in the public, and that, you know, if we know about it, it's in the public, either if it's restricted or we have access, we can have a look at it, or through our regional workshops. So one of the things and benefit of the regional workshops that we do is that it allows us to identify such researchers. And like for instance, I was able to identify several interesting scientists in Morocco and in North Africa, I think last year. And we've now gone on to draft a particular publication on activity. So each time we do the regional workshop, we identify scientists that are doing similar work as well. So to answer your question, yeah, there are efforts, and that is what the workshop is all about. So I don't know, I can just post the paper of the regional workshop so that you kind of appreciate the sort of thing being done, and in terms of the awareness as well that comes with it. So I'm just posting it. So, and just to mention that because of the size of the biodiversity and the size of the project, it's going to be a continuous thing over months, over years, and just keep going. So I just posted the publication, the second publication there.
Okay, thank you.
You're welcome.
Okay, thank you very much. Dr. Omena Bernard.
Thank you. Good afternoon. Thank you very, very much, Dr. Ebenezer, for the excellent work you're doing. I also have two questions. The first one has to do with capacity development. You talked about having training workshops. I don't know how often this workshop is being organized in different parts of Africa, and what are the criteria for selecting those for this particular workshop. Then secondly, you mentioned something about ethics on using the genome of that have been sequenced. I am into research on utilized crops. I'm interested in genomics and bioinformatics.
That's
especially
orphan crops. Yeah.
Exactly, the opportunity crops. And one of them, which I've been working on, is African yam bean.
Okay.
I know it was recently sequenced here.
Sequenced, yeah, we sequenced, yeah.
Exactly. So now I've carried out some in silico study on it, on comparative genomic study. You are interested in the metabolites that are being produced to compare it with other species. Now, having done that, now if I want to publish the work, why have to make reference to, okay, I made it known that the sequences I use were obtained from the NCBI database. Now, if I want to publish the work, is there any ethical issue involved in that? Or do I have to write to the scientists that worked on it to get the permission from them to use that sequence, even though the sequence is freely available on the NCBI database?
Yeah. So your point, Dr. Mena, I'm glad you also mentioned it, because I think Dr. Biba talked about the global biodiversity framework, which is why the meeting was held in Colombia this November, to try to look at what needs to be done. So the discussion is still on. They are going to carry it forward next year and up to up higher. So when you get genomes, when you get samples, right, there are regulations that say you need to do X, Y, Z, and then you sign an agreement with the country or with the person or the institution, right? Before the advent of genome sequencing and next generation sequencing, that used to be sufficient, whereby we sign an agreement. I say, okay, if I come, I take your yam, and then I sequence it. I'm going to train someone from your village. I'm going to build a school. If this yam was gotten from ABU, I know that Abiba mentions it from ABU, then I'm going to build a classroom or do something. It has to be in the form of agreement, right? And in that agreement, you can say, oh, okay, if you make money from it, if it's commercialized, you need to give ABU some percentage or something. So all of those things will be an agreement under the framework of the national regulation and also in line with the Nagoya Protocol, for instance. So, but then when the Nagoya Protocol was established in 2010, not many people thought about next generation sequencing, because as you probably know, issue around ethics and social and policy issues lag behind when it comes to technology. So technology moves very fast. So for example, we had ChatGPT now, before people now started to talk about, oh, let's regulate this ChatGPT because we don't know what it's going to do, right? So that was what happened. So now people are now saying, okay, can we didn't know that you can take your yam and sequence it and get something downstream. Can we also talk about what do we do to it? So countries now, in the meeting that was held in November, two to three topics align with your question. So the first topic is issues around the genome sequence. Once you sequence the genome, it is not a sample anymore, it's a sequence, the digital sequence. So what happens to it? How do we ensure that the agreement that you had with the person in ABU, for instance, is still transferred to the sequence? So, and some countries regulate that through pick and match. So we'll have a paper that will be coming out soon. It focuses on Africa's things, in this direction. Pick and match is what they call the prior informed consent, the material transfer agreement. So in summary, it's just an agreement. But some countries also look into it as a multilateral thing. So multilateral sharing is that, okay, because we don't want to have for each YAML sequence, we don't want to start building agreement. We just put the money somewhere, and then we can get the benefits somewhere. So it looks like there's a noise coming out from.
Okay.
Yeah. So what you need to do is to check with the authors, because normally from the discussion this month, most of the database have been encouraged to put the agreement in the database. So it looks like someone is talking. Can someone mute?
Excuse me, Dr. Odunayo Aziz, please kindly could you mute yourself?
Oh,
yeah. So now, now what you need to do is to check, because now most of these databases have been encouraged to please put the agreement on the data, such that when someone wants to use the data, like just as you mentioned, Dr. Amena, then you can read the agreement and say, oh, this is what this author agreed with this person. And if the person has said, oh, any paper you publish, please acknowledge my institution, University Ahmadu Bello, Nnamdi Ezekwe University of Lagos, then you need to then follow that agreement downstream. So, but it's going to be a long process for that thing to happen. So it's going to take time. But at the moment, you have to just reference the publication or the bioproject ID that comes with it. Is that clear, Dr. Mena? And of course, if you can't, if there's if you have understand what the country wants, that is also on that thing. But it's a bit of a challenge now, because up until this month, no one really does that. No one really puts such agreements on the database, right? So it's kind of a challenging situation.
Thank you.
You're welcome.
Thank you very much. Okay, we still have up to 40 minutes left, 30 minutes.
Mm-hmm.
Yeah. I'm not an expert bioinformatician, and so I really don't know which type of things to, yeah. But as I was saying, okay, we have a Dr. Bentai Benjamin. He's back again. Dr.
Okay,
Benjamin.
thank you, Dr. Aminu.
Yeah.
Thank you, Dr. ThankGod. Dr. ThankGod, just to while away time a little bit.
Mm-hmm.
Yeah.
I wanted to find out how far so far with the Euglena International Network, and is there some sort of collaboration between the Euglena International and the AfricaBP in terms of genome sequencing for the different species of the Euglena, or has anything been done contrary from the recent one I have seen so far? Thank you.
Yeah. So I set up the Euglena International Network, then handed it over since last year or so. So it has a new leadership. So I don't. But at the moment we don't, because Euglena is separate. AfricaBP is separate. So AfricaBP is more Africa focused, and its objective, while its objective is sequencing, we're not sequencing for sequencing sake. So as you can see, we're sequencing to build a much needed capacity to provide the right linkages, and also to do the science and to see how will our science influence policy. Like, for instance, in 2022, I presented at the FAO United Nations meeting, and the AfricaBP work was also useful in influencing the policy, because now, with the Habiba mentioned about the Global Biodiversity Framework, that also align with food and agriculture, genetic resources for food and agriculture. Whatever happens in that discussion at the Global Biodiversity Framework also happens or interact or interfaces with the genetics resources for food. It's called GRFA, because of course, as you know, agriculture is also a biodiversity product as well.
Yeah. And another question from me, Dr. Ebenezer, is that for example, if for something that doesn't exist in my institution or in my geographical location, and I have interest in that, you mentioned that this cannot be a single person's job or a single institution job. There is a need for collaboration, even maybe between countries and also among institutions. So if I have an idea and I present it to the AfricaBP with my proposal, are you the one to recommend, the partner people for the collaboration?
Yeah, so what we'll do is that we'll review your proposal, because we get that a lot. So we'll review your proposal, and then because we have a lot of partners, some of which are also, they come to us, and they also come to us and look at how can they work with us. We'll review it and then find the right partner for you. So we've done that with lots of institutions. I don't know, Benta, if you also saw my tweet today, where the fellows that attend that won the award, they are also in the US. So all of these are through partnership and fellowship opportunities. So yes, we'll review the proposal, we would guide you, we will tell you, okay, actually this is the best thing we've done so far, this is what the current best practice is, but ultimately we will not take the project away from you. We don't do that. We want the project to reside with you and your institution, because we believe that for us to sequence all the genomes in Africa and We need institutions to take more action. We need people on the ground in Africa to take more action. And then if we can work with them to say, generate a very good reference genome, then that reference genome can then contribute to the AfricaBP project. And by doing that as well, the scientists and the institution would, one way or the other, build capacity in their institution. So yes, you send it to us, we review it, and then in most of the time, we usually find a potential collaborator in the project, and we've done that with lots of institutions already.
So yeah.
Okay. Thank you very much.
You're welcome.
Yeah. Hello, Dr. Odunayo Aziz. Kindly. Hello, Aziz.
Finance. Finance.
Dr. Odunayo Aziz. Yeah, maybe he's unaware that he has unmuted himself.
Maybe can continue from there. And because one day to say finance.
Yeah, he's on.
I mentioned when to say business admin.
So don't you have the main?
I don't know. I think Dr. Wadzani, kindly could you should be able to use people on your side. Maybe he's away. I cannot do it here.
Yeah. So but it looks like we've exhausted the questions, right?
I think he's on phone. Allow him to finish. Odunayo Aziz, kindly could you mute yourself, please? Your phone call is interfering with the discussion here. Sorry.
What?
Hello.
I think he has muted now.
Okay. Yeah, you can go ahead, Dr. Ebenezer. Sorry.
Yeah. So yeah, I think I kind of concluded on the question you asked, on the response to the question. So usually we encourage African scientists to send us their proposal. We want the people we work with like 4 or 5 institutions in South Africa, 1 in Morocco, 1 in Nigeria, 1 in Kenya. So most of the time, all of those things come from within. So because we have the expertise, we have the network, we know what the guidance is, and then we work with those institutions to deliver. So we are always happy to get such a request on, this is what you want to do and stuff like that.
Yeah. In fact, I'm very happy with this response. Someone sent me a private message last night. He said they're applying for something, Wellcome Trust, and the Wellcome Trust responded, and they mentioned that they need a bioinformatician in the team. And I think so when this video is uploaded on our website, on our YouTube channel, that person will be able to hear about this conversation, and I think I can provide his contact details. Your contact details. Yeah. I cannot remember the exact number I have. I received hundreds of messages on WhatsApp, but yeah, I will announce that on the platform. I have a Dr. Adrel Echezona.
Yes, good day, sir. Good day, Dr. Ebenezer, Professor Shitu. Thank you for this great conversation. I wanted to suggest, or I don't know if I can ask that, can't we have...
You can go ahead.
Yes,
I just wanted to correct that. I'm not a professor. Yeah, all right. Go ahead, please.
No, well, prophesying that. Okay, so like I wanted to ask, can't we have an organization that helps funding some of this? Or I don't know if we already have something like that that can help funding for some of these great projects that we are having, instead of relying on who we can bring in for applications for other things like that. Is there any way we can pool resources, our own, I don't know, our own personal resources, or somewhere where we can help to push some of our own researches and some of these things that we are coming up with so that it will be able to speed up what we are doing and everything. I don't know if it's really in line with what we are saying.
So I agree with you. If, for instance, that first of all, we know that it's a big problem, and it's not just an Africa problem; it's around the world, even in the US, for instance. And for you to do that, you need to appreciate how gigantic the project is. So that is one thing. It could even take up like sometimes national budgets, right? Of things. That is one thing. It could take on national budget, and that is why it has to work at the institutional level. Most of the projects around the world doing what we're doing is similar model. That is number one. Number two, it's not a project you can do with personal budgets or personal money, because it's huge. It's research, it's science. You don't even know if what you're doing is going to work, right? So so it's not going to. So what I see here is working with institutions, because we also get like resources. We get like people wanting to provide resources from outside, even from within Africa. Our job again is to identify which African institution can we be able to put this into, right? So it's kind of a different model. It's not a typical one grant. If you're going to look at one grant model, then you're looking at like $1 billion, and for most African institutions, that is like, in fact, in most countries, that is like 2 to 3 years national budgets, right? So, but we believe that if you are working at an institutional level, ABU does their bit, Lagos does their bit, University of Nairobi does their bit, then like the scientists in South Africa now, they've sequenced close to 30 genomes, and they were working with us, or they're still working with us, and they started the first sequencing with us, and they're also doing their bit, and they have started to create PhD. They want to create PhD programs in genomics and offer it to African scientists, right? So, except it gets to a point where you have, say, because it's one thing the government can also do, but it's going to take a long time, as you probably also already know in Africa. That's one, and it's also one thing is because it's not something that you say, oh, is generating. So that one way you can do it also is to potentially say, oh, does it generate like direct revenue? So basic sciences, as you do, doesn't always. Sometimes it does, but it doesn't always. It will maybe downstream. When you, just like what Dr. Aminu Shutu was talking about. Let's assume you sequence your genome, then you translate it to the breed, and then you do all of those things, and then you begin to go downstream. How do you now apply it to now increase the yield of the cow milk and all of those things, and then you now sell the cow milk, then that now gives you more money. But you can see how long you have to wait and the whole process. So I think that's one. Just to answer your question, first of all, it's not a single project of 10 genomes. We're talking about thousands of genomes, right? It requires concerted effort across countries and institutions, multi-partner institutions. Majority of them have also have put in resources in AfricaBP, and some of them have also contributed, both industry and the public and private sector, both in Africa.
Sorry, Dr. Ebenezer, sorry for interjecting, and this is the last question, please. I wanted to ask, in your own view, do you think all the institutions are properly collaborating? For example, there is this institution, African Center for Genomic Excellence, that is in Osun State under Redeemers University, that kind of won a huge funding from World Bank and everything. Are we properly synergizing and working properly to be able to, do this sequencing of genomes and everything properly? Do you think we are actually working properly, collaborating properly within our own region? I'm not
West
talking
Africa.
Yes. Are we properly collaborating, or we still have some people that are not really fully involved in all of this? Because, for example, the HG, for example, the African Center for Genomic Excellence, they have a lot of this next gen sequencers, different things like that would really help speed up the project. So I just wanted to get from your own perspective. Thank you.
Yeah, so I think from in other regions like South Africa, Southern Africa rather, there's huge collaborations as well. But because Africa is divided into 5 regions, and collaboration speed will differ from each region, and that also is a reflection of the technologies that are there. So with SG, I know the founder, and we also communicate. He also organized practical workshop. We partnered last year is it last year or this year? I think it's this year. So to organize a practical workshop in Institute for African Scientists. I think we trained like 10 to 15 people. So what another thing you need to look into is like some institutions are into health, for instance, or more into health and maybe moving in other directions. What we are looking at more is biodiversity, sequencing biodiversity for conservation purposes, and other aspect of things. So while I do know that, for instance, in West Africa there are activities going on, there are also opportunities for more things to be done, and all those things also depend on priority. As I mentioned, if, for instance, you have an infectious disease center, it's going to take time for them to again be able to sequence all of those things. So I think one thing you need to know in biodiversity in Africa is that there hasn't really been biodiversity efforts in Africa up until 2020 when we started AfricaBP. So our effort has been to provoke action, work with institutions to say, okay, can we start sequencing this? Can we start moving in this direction? So it's going to take some time, but gradually we're going to move fully. Before we started, I don't think there has been any national effort or even local effort on, say, sequence African biodiversity. No, there hasn't. But as I speak to you now, South Africa just initiated their own national biodiversity project, and it was inspired by the AfricaBP, and they are sequencing 1,000 genomes, for instance. And Morocco is also thinking about the same thing. They are anchoring it onto national agenda. So for us, it's all about provoking those action. And one way I see that your question could be answered, Adriel, is maybe from this meeting, is those in West Africa and Nigeria can say, okay, can we go and start up our own Nigerian 1,000 genome biodiversity project? And that could help people like Habiba, for instance, and knowing that at the national level, there is already an activity that everyone could fit into, right? And what would then happen is you don't need to worry about West Africa. You start at the national level, just as we were able to inspire South Africa to start. Now they have like the 1,000 project. All the scientists are on the it, and Morocco is trying to do the same. Now the guy is trying to. I think we spoke a few weeks ago on this effort. So if there's one activity or one action point from this meeting is, I think Dr. Aminu Shitu giving you that challenge, is you all go and say, can Nigeria come up with its own you just start small. You don't need to do 10,000. Just say 1,000 genome projects for Nigeria, focusing on plant and animal, and then say, okay, can you work with the African BioGenome project? We don't interfere with what South Africa is doing at the national level. What they do, they connect with us so that we're that umbrella. We're kind of an umbrella platform, and everyone can now come and do. So I'm hoping that's a yes from Dr. Aminu Shitu and Dr. Biba and Andrea.
Yeah, I think that's a good idea. It's a big challenge. Easier said than done, but we'll look at it. Since you're around to offer some help, fine. Thank you so much.
You're welcome. Bentai?
Dr. ThankGod for this provocating idea. I think we can leverage on some of the national institutions, like the NAFDA agency.
Exactly.
You understand? All you need to do is put up a team, a very simple team of like 5 researchers, come up with a very good proposal, and then go and meet with them. This is what we want to do, because I discovered one of their major challenges over time is funding. most of the people over there, I discovered they are lackadaisical about the future in biotechnology, in genomics, in bioinformatics. But if you can bring up an idea, since we have an African institution who is ready to spearhead capacity building, all we need to do is put up the idea in form of proposal. It must not be an excellent proposal, a very simple write-up, you understand, that can be reshaped over time. You understand? This can be achieved.
Yeah. So
1,000 is not a big deal. 100 is not a big deal. 500 is a good start.
yeah, exactly.
Across maybe zonal, maybe
Exactly. So you can say there are 6 zones in Nigeria. You can say,
Exactly.
so just to clarify, do we have more Nigerian researchers here? So like we don't feel like we're making this in
Almost
Nigeria.
everybody is a Nigerian.
Okay, yeah. So my suggestion to you, to the leaders of this group, I don't know who is the leader, but I think it's Dr. Aminu Shitu, correct me if I'm wrong, is from this meeting today, and that is how other regions started, like South Africa now, you can Google South African 1000 project. It's what inspired by AfricaBP. What happened is that they went, let me just give you, they had like a national genomics thing, and they said, okay, the national genomics things can anchor it, let's connect all the national infrastructures to it. So you can, in Nigeria, there are 6 regions. You can say, okay, we want to sequence 500, depending on the number you have. You cannot divide, let's say you are going for 1,000. You can divide the 1,000 by 6 and say, okay, in each region of Nigeria, I don't know, 1,000 divided by 6, how many will it give you? Say 200, 300, something like 150 or something. You can say, oh, each region take 150. Then in that 150, you can say, okay, we're sequencing this XS number, in each region. We're sequencing 150. Then in that 150, we're going to sequence plant 50, 40. We're going to sequence animal, microbe, based on that. And the national infrastructure, each region in Nigeria has a national biotech infrastructure, or even genomic. Andrea just mentioned, for instance, in Osun State, we have the SG there. You can say, oh, that one and any other infrastructure there can take care of the 150. You might think 150 is small, but as you start to sequence it, you see there's a lot of effort. It can take you a year or 2 or 3 years to actually, even sequence that small number. But what you appreciate after 1 or 2 years is the things that you've done, the genomes you sequence, how it has provoked effort, how it has trained people, how it has, more publications. Like all the scientists now that are part of AfricaBP, they've also been authors and things. So all of those things are helping their career. So yeah, that is how I would suggest it. First of all, list out in each region which are the top biotech research centers that can do this, and put up a proposal, as Bentai said. Then someone has to drive it, and it has to be someone based from within Africa, or from within Nigeria rather. And if your network puts that is also a good thing to do.
Yeah, I think one of the purpose of the AfriScience network is to link up researchers and collaborators. I wish I like fixing webinars on Saturdays when people are at home. I think the maximum attendance for this webinar is 31 people, which keeps on fluctuating. Now we have 27 people with us. But I believe that when this discussion is uploaded on our YouTube channel, I'm going to advertise it very well. And when people listen to our conversations, and maybe they will start thinking on what to do. I wish this discussion is on the weekends where we have a lot of people for brainstorming, but notwithstanding, it is quite excellent. People are busy in the office. I can see Dr. Oko, Edith. You have something to share with us, please. Hello, Dr. Oko, Edith. Are you there? You are still raising your hand. I want us to spend the last 13 minutes. Okay, she has dropped her hand. All right. Dr. Habibat, any more insights from your place?
No, nothing. I'm just looking forward to the collaboration he mentioned, Dr. Ebenezer mentioned. I'm really looking forward to it. And thank you once again for the wonderful presentation.
All right,
You're
thank
welcome,
you.
Dr. Abiba. Yeah. Bentai,
Yeah.
seems like you have another question.
Okay, Dr. ThankGod, is it possible when you want to start an initiative like this to write the African BP for maybe to borrow or maybe to share some of the successful
Materials.
African materials like
Yeah.
their proposals, you understand, so that it will
Yeah.
serve as a reference material, as a guide for you
Mhm.
to initiate yours. Like
Yeah,
the South
so
African
the South African people, they came. They also had support from, they had things because they were part of us. Morocco, we also shared. So I can share something with you. Let me see. I can very quickly. That was a good one. Thanks for raising that. So I can share something with you that other regions and countries are using, and so this is the link. This could be a good start. So you don't need to do it exactly, but it should give you idea. What I just sent it in the chat box.
Okay, all right. I think
So it should give idea. It should give you idea what how other people are using it. So this is free to use, and on our website you can also see things you can use, like I'm just going to post documents. So feel free to borrow it. Just what you just need to do is look at the acknowledgement that needs to go with it, or when you do use it, because AfricaBP is for all Africans. So would appreciate if you just say, reference the if it's a publication or anything like the document, reference it or reference AfricaBP, so that the people who develop this can also be acknowledged. So I am going to just post like another document that allows you to, so you can use this as a reference starting point. And I just posted it. I just posted this one as well. So you can use that one. So maybe, yeah, that could be a starting point. You don't have to do it verbatim or you don't need to do it verbatim or exactly, but just gives you an insight on how to approach it. So other countries have used the same.
Yeah, so I have 5 documents to share on the AfriScience network platforms.
Okay.
So I think, shall we round it up?
Yeah.
10 minutes to the end of time. And I would like to thank Dr. Ebenezer on behalf of everyone here, on behalf of the AfriScience Network, all the administrators of the AfriScience Network, 7 of us. I extend our heartfelt thanks to Dr. ThankGod, Echezona, Ebenezer for such an engaging and insightful presentation on the African BioGenome Project, the progress and perspectives. Dr. Ebenezer, your expertise and dedication to advancing genomics and bioinformatics in Africa have truly inspired us today. I have learned some new things. The depth of knowledge that you have shared and the remarkable achievement of the AfricaBP highlight the transformative potential of this initiative for the science and society. And I must say that we are highly grateful for the time and effort that you devoted to being with us today and for enlightening us on this important topic. Thank you very much once again, Dr. Ebenezer, for an excellent presentation. And we look forward to working with you in the future. Thank you so much, sir.
Thank you, Dr. Aminu Shitu, and to everyone. And yeah, I look forward to future collaborations as well.
All right. Thank you.
Bye.
Thank you. Thank you for the wonderful presentation.
Thank you, Dr. ThankGod.
Thank you, everyone.
Thank you very much.
Yeah.